oversimplify it, this hairpin loop is the reason why the RNA polymerase falls off the template DNA and you end transcription because of this. So those are the two ways prokaryotic transcription is terminated / prokaryotic transcription is ended. That is row-dependent and row-independent termination. Transcriptional regulation in prokaryotes. Lots of you did really well on the extra credit. That's good, because you may be given several multiple choice questions asking about the lac operon. Okay? We know there are two components to regulating the lac operon. Oh, I don't like these diagrams, but you know them. You've all drawn them for me. I've had to read all of your extra credit questions. The first way the lac operon is regulated is via what mechanism? Negative inducible, right? You may be given questions about that, such as what are the co-inducers of that? How does that work? What about the second part? What's the second way a lac operon is regulated? Positive inducible via an inducer called what? Cap, right? You may be given questions asking about that. You also even told me, lots of you told me on the extra credit, what happens when you have lots of lactose, or lots of glucose, or not a lot of either of them, or one or the other. You told me what happens in each of those situations. You may be asked about that on the exam as well. So I expect nobody to get anything wrong regarding the lac operon. You guys did very well. You guys made me great things all night long for that extra credit. So I expect everyone to do well on it. You are welcome.
John, you said that if we are asked how the lac operon is regulated, it's both negative and positive?
Yeah, good question actually. The lac operon technically, if the question is only how is the lac operon regulated, technically the answer is the negative inducible part. Because the cap stuff is secondary, that is bonus stuff. Okay? So if it's asked about how the lac operon is regulated, just by itself, that's all it's asking about, negative inducible. If it asks about cap, you talk about cap and how it affects the lac operon. Okay? Yes?
So we won't be asked, what is the secondary, and then just say positive?
No. You learned already enough about this. I'm sure you guys will answer it very easily. You won't be asked collectively. It'll be separately. I haven't run into CRISPR yet, right? Okay, so let me skip that for now. Okay. All right. I guess it's in 29B, so excuse me.
Do we know about HIV?
I will give you, so about HIV, there will be stuff about HIV on the exam. However, I'm going to give you guys that as separate information through email. I'll give that separately apart from this review, because there's specific information you need to know about that that would take a bit too long to explain. So I'll give that separately. So, eukaryotic transcription. Now we get to material that was not asked about on exam 3 that you're going to need to know for the final exam. We have reviewed a lot of this at SI already, so I will only highlight some things. That is the fact that eukaryotic transcription is very complicated because there are several different regulatory sequences and several different regulatory proteins called transcription factors. The regulatory sequences, we know there are 3 categories: core promoters, regulatory promoters, and this extra thing that's really unique for eukaryotes called enhancers. Enhancers are regulatory sequences that are, as you can see, very far away from the gene that is actually being transcribed. This enhancer sequence, and technically any regulatory sequence, is where transcription factors will bind to. Okay? There are 2 categories of transcription factors when we talk about eukaryotes: general transcription factors and specific transcription factors. General transcription factors will be involved in all eukaryotic transcription. General transcription factors such as TF2D, you don't have to memorize them, but general transcription factors, they work by binding to the core promoter only the core promoter. When they go to the core promoter, they recruit the machinery needed to transcribe a eukaryotic gene. However, in addition to the general transcription factors, we have specific transcription factors. These things can bind everywhere, but they are specifically unique because specific transcription factors are the only ones that can go to enhancer sequences. Okay, so general transcription factors only go to the core promoter region. Specific transcription factors can go everywhere, but what's really cool about them is they're the only ones that can go to enhancer sequences. So special transcription factors, or specific transcription factors, go to enhancer sequences. Collectively, the mixes of general and specific transcription factors regulate the expression of genes. So when you look at one gene versus another, depending on which transcription factors are involved, affects how highly or how low the expression of that gene ends up being. And one such feature that is illustrated at times is the following. I have to find a slide illustrating this.
Transcription factors are only found in eukaryotes, correct?
Yes, transcription factors is a eukaryotic thing only. So, that enhancer stuff I was talking about. Transcription factor binding to enhancers is one of the features that gives you this different expression of genes in one tissue versus another. This slide attempts to illustrate that for you, so I suggest you review this in detail later, but a short explanation of it is when you look at liver cells, they have certain transcription factors that are liver specific and certain enhancers that are active in the liver to express liver genes. When you go look in the same organism, but you look somewhere else, such as in the lens has lens-specific transcription factors and lens-specific enhancers that are active to express lens-specific genes. So this specific transcription factor and enhancer relationship is what causes these different genes to be expressed in different tissues, even though everything comes from the same organism. Oh yeah, this thing. This cool feature, DNA bending protein, you can see it is only something you only find this in eukaryotes. The DNA bending protein is something you only find in eukaryotes. And that's because you can see what is illustrated here is that that's the only way for enhancer sequences to be brought closer to the transcription start site is via a DNA bending protein.
You said eukaryotes?
Yes, for eukaryotes only. Now, you only see that in eukaryotes. Eukaryotic RNA polymerase. This specific one, RNA polymerase 2, is responsible for transcribing mRNA. You may notice a few features of this polymerase that sound very similar to prokaryotic RNA polymerase. In particular, this is written here, the fact that eukaryotic RNA polymerase has two subunits in there, RPB1 and RPB2, that are homologous to prokaryotic RNA polymerases beta prime and beta subunits. So make sure you compare those two things to each other. Find the similarities between them, between eukaryotic RNA polymerase and prokaryotic RNA polymerase. I can't remember where it's illustrated, so I will describe the following. rRNA is a special type of RNA that is made by one of your eukaryotic RNA polymerases. You'll have to go find which one. I believe in you guys. rRNA is made by one of the three RNA polymerases in eukaryotes. What's unique, especially, is that rRNA is made in a particular area in the nucleus called the nucleolus. You might remember that from Bio 1, right? The nucleolus is this really specific part of the nucleus. We now know in our class that's a specific area where rRNA is transcribed, okay? Where rRNA is transcribed. It's in the nucleolus. That's a very specific detail you guys should know. Okay. So I hope CRISPR's in here somewhere. I think it's, yeah, here we go. You may be given a diagram such as this, where nothing is labeled, and you may be asked to to identify where on this diagram you find the guide RNA and the PAM sequence. Those are the two important labels on this diagram that you should know about. So you may be given this diagram, you may remove the labels, you need to point out where the guide RNA is on here and where the PAM sequence is on here. Overall, you guys remember in exam 3 what CRISPR is useful for, is to do what to a gene? To knock out a gene, to remove a gene from the genome. Okay. The next bit of information I have already reviewed during the Monday SI session, so I posted at the same time as this review. I won't go over it in detail, but I will highlight some things from the stuff I went over on Monday, and that is the following from 30A and 30B. Is this stuff about codons. I talked to you guys about what happens if our genetic code is not a triplet code, but is instead a duo code. You had to mathematically calculate what would happen with that. What would happen if the code, instead of being a triplet code, it is a quartet code? You had to calculate what would happen in that case. That was all illustrated earlier. I'll post it at the same time as this exam review. We learned about some vocabulary words that are associated with the genetic code, such as, right here, non-overlapping and continuous. There's some definitions associated with those. And the most important word associated with the genetic code is the phrase degenerate code, or degeneracy. This I will actually explain using this slide. Our genetic code is a degenerate code because some amino acids can be coded by multiple codons. Multiple codons can code these amino acids. That phrase, that is defined by the word degeneracy or degenerate. One amino acid, multiple codons. In 30A, we talk about a particular enzyme called aminoacyl-tRNA synthetase. The name of this enzyme gives an idea of what it does. Its job is to take tRNA and take an amino acid and put them together. It synthesizes an amino acid to a tRNA. It specifically does so by attaching it to the 3' end of that tRNA. It attaches it to the 3' end of that tRNA. This enzyme is pretty special because of a couple different features. Let me make sure I don't forget to tell you. This aminoacyl-tRNA synthetase is able to proofread itself. So similar to the DNA polymerases that we learned about, this thing can also proofread itself using this editing function called deacylase. So what I mean by that is sometimes this enzyme puts the wrong amino acid onto the wrong tRNA. It has the ability to fix itself by removing that amino acid and trying again. And that removing part is done via deacylase. Probably not listed here, but talked about in class, is that this enzyme is very conserved in terms of evolution. We see this in all types of organisms, prokaryotes, eukaryotes, archaea. It is a very conserved old evolutionary enzyme, aminoacyl-tRNA synthetase. I don't know if that's stated in the PowerPoints, but it's talked about in class, so I'm giving that information to you here as well. It's very old evolutionarily speaking. Wobble base pairing. I've gone over this also last week and some on Monday as well. Wobble base pairing is based off the following basic concept, of which you will have to apply some things to it, is that during translation, where you take mRNA and you translate it into proteins, you must bring in tRNA to match your mRNA. You do so 3 bases at a time. So on mRNA, those 3 bases at a time we each call codons, 3 bases at a time here. The third base on your codon and the first base on your anticodon are in a position called the wobble position. The wobble position is special because it's the only place where you can have non-canonical base pairing, meaning unexpected base pairing. Some examples of unexpected base pairing are G can sometimes bind to U, and so on. I've given, on Monday's SI session, I talked about ways to use the codon table to answer particular questions, and I will post that also with the exam review. You may have to use the Wobble table, I should say, to answer particular questions. Yes?
We are giving the table.
You will be given the Wobble table, the codon table, all the tables that have been used and were covered after exam 3. You will be given for the final exam. Oh yeah, because you'll be given a codon table, you may be given an mRNA sequence, and you may be asked to give the amino acid sequence that is translated from that mRNA sequence. Remember 2 important features of that. 1, you do not begin translation until you are at the start codon. So you may be given a really long mRNA sequence, but part of it you may ignore because you must begin translation at something called a start codon, which is this thing right here, AUG. You must also end a translation. You must also stop sequencing mRNA into protein at a stop codon. So if you're given an mRNA sequence, give me the amino acid sequence that is generated between the start codon and the stop codon. So there are some unique features of the genetic code that are illustrated, and I believe it's not this slide, this one, is that the stop codon UGA can sometimes actually translate into an amino acid. That's specifically detailed to 2 exceptions to the genetic code. Is that this stop codon UGA can sometimes be used to code for tryptophan, and sometimes it can be used to code for the 21st amino acid called selenocysteine. Okay, so that is, both of these are coded specially by the stop codon UGA. Yeah. Okay. I believe there's a vocab word out here I forgot to point out. That is the phrase synonymous. Synonymous codons. Synonymous codons are codons that, when you go look in the codon table, 2 codons are synonymous if they code for the same or similar amino acid. What I mean by similar amino acid is 2 nonpolar amino acids or 2 acidic amino acids. Those are similar amino acids to each other because of the same chemical type. So synonymous codons are 2 codons coding for either the exact same amino acid or chemically similar amino acids.
They have to be nonpolar.
No, it could be as long as they're the same chemical type, that would still count as synonymous codon. So either 2 acidic amino acids would be similar to each other. All right. Let's try our luck today. Oh, that's a good sign. All right. So, I will not cover everything, but this is a great animation. If you guys don't know yet, in 30B, there's one animation that basically covers 99% of what you need for 30B. That is found on slide 4 here. You take this web address, plug it in here, and there's this animation here describing prokaryotic translation, the steps of prokaryotic translation. I'm going to jump to the steps in detail.
So basically, all we have to do is just look at that video instead of like looking at the PowerPoint.
Yeah, the video, the animation covers covers everything for prokaryotic translation. For eukaryotic translation, you do have to go to the PowerPoints for that. All right, so initiation is the first step in prokaryotic translation. It requires a few materials. It requires this pink thing, the small ribosomal subunit. It requires the mRNA template. It requires some factors called initiation factors, of which there are 3 of them. It also requires a couple other things. It requires an initiation. To begin translation, you need the very first aminoacyl-tRNA, which is always going to be a tRNA holding on to methionine. And when we're talking about prokaryotes, as we do most often, what is special about this methionine? It has a formyl group attached to it. Additionally, we need GTP to power this thing. So we have small ribosomal subunit, mRNA, initiation factors, formal methionine on a tRNA, and some GTP. To signal the end of initiation, we need the large ribosomal subunit to come in. So we need this to finish off initiation, and then all those materials I have listed are necessary for initiation. Notice it comes in, GTP is hydrolyzed, we throw away the initiation factors because we're done with initiation. We've now assembled the ribosome. Skip to we talk about elongation. Okay. General stuff about elongation. I detailed this in detail last week. Elongation is a cyclical process where we bring in a new aminoacyl-tRNA into the ribosome. This is done via the elongation factor called elongation factor TU. Notice that it hydrolyzed GTP in order to bring in this amino acid. In order for this elongation factor to bring the next amino acid, it has to regenerate itself. And that's illustrated on here. I'm going to have to skip it because we're running out of time. But it's slowly regenerating itself. Now look at that. It has a brand new GTP. It's going to pick up a new amino acid so that I can bring it to the ribosome as well. Simultaneously, whatever the elongation factor is doing, in the ribosome, the large ribosomal subunit is the one that is fusing together these amino acids. The large ribosomal subunit is the one putting amino acids together, forming peptide bonds. You don't have to worry about this animation, so let me move past it. Okay. So they're going to show this several different times. The dark one? Yes, the dark purple one is the large ribosomal subunit. Okay, every time a new amino acid tRNA is brought in, everything shifts to the left so that you can bring in more amino acid tRNA. So you can play with that animation, but you see that elongation is bringing in new stuff, fusing it to the growing peptide chain, and shifting everything over to the left so that you can read the next codon. Repeat it over and over again. All right, so after you do that for a while, notice it's reading one codon at a time, bringing in a new amino acid one at a time, until it reaches not a regular protein coding codon, but something called a stop codon. Instead of a tRNA being brought in, what happens at the stop codon is you have release factors bound to this. When release factors bind to this thing, it will initiate the termination step in translation, which causes the large ribosomal subunit to become a hydrolase. When it becomes a hydrolase, it's going to cut off this amino acid sequence and it's going to float away like that. Okay? So the release factors cause the large ribosomal subunit to become a hydrolase, to cut off the amino acid sequence and end translation. All right, last few bits of information you will need is the eukaryotic translation stuff, and that is listed in one nice slide for you. These two slides here, where you have the numbers 1, 2, 3, 4, 5, 6, are all the differences between eukaryotic translation and prokaryotic translation. You only need to know the first 4, okay? There's no time to go over it, but for eukaryotic translation, you need to know the differences between eukaryotic translation and prokaryotic translation. They are listed in the first 4 points here at these 2 slides. And lastly, and again, it is jumping around. Let me go to the codon table to illustrate this, because I don't remember where it talks about this. You can have mutations that don't actually cause a change in amino acids. So, for example, let's say you originally had a codon that is CUU. A mutation caused it to transform into CUG. Normally, you might think that's not a big deal. It used to be leucine at CUU, and the mutation turned it into CUG. It is still leucine. That is called a silent mutation, where a mutation does not result in a change in amino acid. That is called a silent mutation, that you can't see it at the protein level. That is not always harmless, however. Sometimes that can be harmful, because each of these codons, some of them are faster than others. What I mean by that is when a ribosome is reading one codon and the next codon and the next codon, some of them it reads a lot faster than others, which it reads slower. So for example, I don't know if one is fast or one is slow here, but to illustrate as an example, let's say CUU was a fast codon and CUG was a slow codon. This mutation, although you didn't change the amino acid, the act of switching the speed, what the result of that is that you end up producing this protein a lot slower than you normally would, because you've switched from a fast codon into a slow codon. That is an example of when a silent mutation can sometimes be harmful. All right, so that's all I will talk about now. There are 2-3 other things I will give you guys in detail through email because it's hard to explain on here. Please review the old exam review videos for more details. I'll be out there. Yeah, thank you.